Hello. Welcome to 3 Minutes with QIAGEN, where you get quick answers to your specific questions in just 3 minutes. Firstly, please take note of our legal disclaimers. QIAGEN products shown here are intended for molecular biology applications. These products are not intended for the diagnosis, prevention, or treatment of a disease. Today's question is, when should you use targeted DNA sequencing? Let's ask Rayed Samara, Global Product Manager for Next Generation Sequencing Technologies at QIAGEN. Rayed?
Thank you, Ali. Targeted DNA sequencing should be used when one wants to achieve any of the following. First, focus sequencing capacity on certain genes of interest, which saves time and money.
Can you give us an example?
For example, if one is interested in identifying the role that certain BRCA1 mutations play in breast cancer, one can focus all the sequencing capacity on BRCA1 itself without the need to sequence the entire genome. And second is to do deep sequencing, which identifies low-frequency or rare mutations.
Which types of research would this apply to?
For example, cancer research. So it is known that tumors are very heterogeneous samples consisting of several cell types, which results in certain mutations being present in a very low number of cells relative to the entire population. The identification of these low-frequency or rare mutations can be achieved very efficiently by targeted DNA sequencing. And third is to increase depth of coverage, which increases confidence in sequencing results.
How does depth of coverage translate into increased confidence?
Next-generation sequencing has higher error rates compared to Sanger sequencing, so each base has to be sequenced many, many times in NGS, a parameter known as depth of coverage. The higher the depth of coverage, the more confident one is in sequencing results. This can be easily achieved with targeted DNA sequencing. And fourth, sequence limited amounts of DNA starting material, such as from FFPE samples, fine needle biopsies, and laser capture microdissection samples.
Why is targeted DNA sequencing so good for FFPE samples?
FFPE samples usually yield very limited amounts of DNA and low-quality DNA, which limits their capability to be sequenced. Targeted DNA sequencing, due to its lower requirement for DNA material, makes sequencing FFPE samples a possibility. For targeted DNA sequencing, QIAGEN has developed the GeneRead DNA-Seq gene panels. These panels allow you to sequence the 20 genes that are most relevant to the disease you are studying. For example, the GeneRead DNA-Seq breast cancer panel allows you to focus on 20 genes that are known, based on current literature, to be altered in breast cancer instead of spending your sequencing efforts on the entire genome. For additional information or special trial offers, please visit us at www.qiagen.com or contact us at the email shown here.